I'd like to start by thanking longevity leaders very much for inviting me to give the talk, which is going to be about the role of a virus in Alzheimer's disease. This is a very sinister virus. It's really, I think, at least as dangerous as the coronavirus, but is much, much more insidious. And so it's very difficult to explain how it is really, well, you'll see from the data how serious it is. But we have to start off by thinking what causes Alzheimer's disease. And there are only a few risks that are known. First risk is age. Down syndrome is another risk, family history of dementia, which applies to only a very small proportion of cases, maybe a few percent of Alzheimer's cases. Head injury and the type 4 form or allele of the gene for apolipoprotein E, which I haven't got time to describe now. But none of the above factors is either necessary or sufficient to cause the disease. So in each case, if it's operating, another factor must be needed to act with it. So let me describe briefly virus characteristics for those who are not too clear about them. Well, their main aim is to replicate, and they can do that only inside cells using the cell machinery, and that usually kills the cells. However, herpes viruses are a class of viruses which can remain in the body lifelong, so that some of them that don't kill the cells can become latent, in other words, dormant, and they probably cause little harm, although they can be reactivated to an active state by events like stress, immunosuppression, inflammation, etc. Now, I would like to stress the fact that the effects of infection vary from asymptomatic to fatal. In other words, infect does not necessarily mean affect, as controls too can be infected. In other words, they're asymptomatic, and that might depend on host genes as well as other factors. This is an important concept because I'm going to be showing you that the proportion of people with virus in brain is as rough, almost as high in controls as it is in Alzheimer's patients. This does not mean that the virus is not a factor. This is very commonly thought to be the case that if somebody is infected, if a group is infected, another group isn't affected, then the infected ones must be the ones that show symptoms. But in fact, many cases are asymptomatic. And perhaps the best example of this is tuberculosis, where 10 times more people carry the tuberculosis bacterium than actually develop TB. So only 1/10 of those people infected actually get the disease. And that's a very good example. Same applies to herpes, which causes cold sores. Herpes simplex type 1, the usual cause of cold sores, or herpes labialis. It affects something like 25% of people who are infected with the virus, but the rest are, although infected, are asymptomatic. Now, getting on to damage. On reactivation, where the virus becomes active, the damage happens through direct virus action and also through inflammation. Now, why is a virus involved? What are the arguments for that, and what are the arguments for herpes virus being involved? Well, several lingering viruses, for example, measles and HIV, can cause neurological disease long after primary infection. Herpes simplex type 1, HSV1 in short, is a likely agent in Alzheimer's disease, as firstly it's ubiquitous. It affects most people when young, and certainly some 80% or more by the time they're 60. So it goes on infecting people, and these days where socioeconomic conditions are better, as in the more advanced countries, it tends to infect people at an older age, but in the past it was babies who were infected after being kissed, rather wet kisses from carrying saliva from a loving auntie or parent. So it infects most people. Also, it can reside lifelong, as I've mentioned, in latent form in peripheral neurons, but it can reactivate often, as I said. And in some people, about 25% or so, it causes cold sores or herpes labialis. And the third reason, that in the very rare acute brain disease, herpes simplex encephalitis, short HSE, the regions affected are the same as those affected mainly in Alzheimer's disease. So how did we start the work? What were the questions we asked? Well, we firstly asked, obviously an important question, is the viral DNA present in normal brains? And obviously, if it's present, it's much more likely to be an agent. If it's absent, it could possibly be an agent, but it's less likely. So we wanted to look. There had been a moderate amount of work in the 1980s looking for HSV1 DNA, but the methods used, hybridization methods, were relatively insensitive and could detect only something like, at best, one viral genome per cell or more, which is obviously a very high level of infection, a ridiculously high level. So we use PCR, which is vastly more sensitive, and that was the first study using PCR of infectious agents in brain. The second question we asked, if the virus DNA is there, is the virus active in brain or is it just a passenger? And next, if it's in brain, is it associated with the features of Alzheimer's disease, AD? And lastly, and most difficult to answer, is there a causal link between HSV1 and AD? So to show you what we started off with the first one, is it present in normal cells? We looked by PCR for a sequence in the viral DNA in brain, and we looked in 61 specimens, sometimes 2, sometimes 3 specimens from frontal and temporal lobe, occasionally hippocampus, in 61 people, subjects, 3 samples or 2 samples from each brain, 61 Alzheimer patients, 48 elderly normals, and 28 young normals. And what we got was, this is the detection frequency, the proportion of people who had virus in brain. And as you can see, the values for the Alzheimer's and the elderly normals are similar, 70% and 60-something percent. There's not statistically different, and we've never had enough funds actually to look at a much larger number, which would tell us if there is a true difference. Whatever, even if there is a difference, the value is remarkably high for elderly normals, and also very low, 10% or so, for young normals. So this was quite a striking finding, and we were, I have to admit, very disappointed because quite naively we'd thought or hoped that maybe there'd be virus in Alzheimer brain, but none in age-matched normals, but that wasn't the case. So the next question we asked, well, is there a difference in the group that is the Alzheimer's disease group? In some respect, is there a difference between that group and the elderly normal people? And what we decided to look for was who was carrying the ApoE4, the allele of ApoE, which was known in the 1990s. It was found that it was known to be a risk factor for Alzheimer's, although not an independent one. It had to act with something else. So what we did was we looked for the ApoE frequency carriage of HSV1 positive and negative AD patients, and as you can see, the proportion of the Alzheimer patients who were HSV1 positive, 45 was over 50%, a very low value, only just over 10% in the HSV1 negative. Then in the elderly age-matched normals, the values were right down about 5% and 8%, I think, in the HSV positive, HSV negative. So what this shows was a very strong association between HSV1 positivity and a carriage of an ApoE4 allele. Now, I had a crazy idea and thought, well, we'd look at what the frequency of ApoE4 was in people who were suffering from herpes labialis, cold sores. So what we did was we looked at the allele frequency of a group of cold sore sufferers and compared those with non-cold sore sufferers. I think there was something like, not very large numbers, but nearly all were HSV1 positive. So nearly all of them were infected with this virus. And what we found was a striking, again, association of ApoE4 frequency, 30% in the case of the cold sore sufferers, and a much, much lower proportion in the case of non-cold sore sufferers. So again, a strong association, which is really very nice support for a thought that ApoE4 and HSV1 in brain together confer a strong risk of Alzheimer's disease, because this is looking at herpes labialis, you're looking at the peripheral nervous system. And what this tells us is there is a strong association, which could well be a risk, or we don't know that, for herpes labialis if you carry an ApoE4 allele, herpes labialis being caused by HSV1. So the two together confer this risk in the peripheral nervous system, so it is very much parallel to what happens in the central nervous system, with the two conferring a risk of Alzheimer's disease. In fact, we were able to argue in our Lancet paper in 1997 the two together were likely to be causal rather than just an association, but I haven't got time to go through that now, obviously. Then the next question we asked, is the virus active in brain? Because it could just be a passenger doing nothing really, or just sitting there quietly. And I'll show you in the next slide what we did. We looked for antibodies against HSV1 in the cerebrospinal fluid. The argument, the basis being that these are known to persist for years after herpes simplex encephalitis. And what we found was the antibodies in the CSF of over half the Alzheimer patients and the aged normals. So in other words, these are confirming the presence of HSV1 in brain in at least half. In fact, these values are lower than the values of the HSV1 DNA we found in brain, but we could easily explain that. Again, I don't have time to do that now. But we did a careful check that this was not due to leakage of these antibodies from the blood across the blood CSF barrier. It was not, though. And also, we looked into a few cases of infant CSF, cerebrospinal fluid, and we found no antibodies in those, which is consistent with the fact that only a low proportion, very low proportion of young people and infants actually had viral DNA in brain. So we conclude from this that HSV1 in elderly brains has reactivated, perhaps recurrently, causing an acute infection and therefore damage, considerable damage. And this damage, we argue, is cumulative. Reactivation events probably occur repeatedly, and each time perhaps not at a very high level and perhaps localized, but in some causing sufficient damage to lead to Alzheimer's disease eventually. So we then went on to ask, is there a causal link between HSV1 and Alzheimer's neuropathology? The main features of the Alzheimer's neuropathology, as you probably know, are amyloid plaques and neurofibrillary tangles. The amyloid plaques are regions of the brain which contain a high proportion of this peptide amyloid, and the neurofibrillary tangles are, as you say, fibril-like tangles consisting mainly of abnormal form of the protein tau. So what we did was we stopped using brain because obviously you couldn't do experiments with cells from post-mortem material. We looked at cultured cells in culture, which we infected with HSV1. We looked in this case at human glioblastoma cells. These are brain type cells. And the lower slide on the left-hand side shows uninfected cells. The upper one shows increase in staining using immunocytochemistry, specific antibodies to amyloid. That amyloid A beta is produced or accumulates in culture cells after infection. On the right-hand side, we're showing uninfected mouse brain at the lower right-hand side, and top right, we looked at some specimens of HSV1 infected mice, looked at their brains, and as you can see, there are deposits there which are very similar to the deposits in HSV1 infected cell cultures. And these were looked at by immunohistochemistry, again using specific antibody to beta amyloid. We decided also to look for abnormally phosphorylated tau. And in this case, we used both neuroblastoma cells, which are a brain type cell from humans, and the glioblastoma cells. And the top left shows uninfected cells, and the bottom left shows HSV1 infected human neuroblastoma cells. And you can see there's a great deal more black, in other words, showing that in accumulation of phosphorylated tau after infection, both in top being uninfected in both types of cell, each type of cell, and the bottom being the infected in each type of cell. In this case, there is some staining in uninfected cells. That's perfectly explainable, and we did explain it in our paper, Wozniak et al. 2009A. But again, I haven't got time to go into explain that. But anyway, as you can see, the increase in deposition of this abnormal phosphorylated tau is much greater after HSV1 infection. And next, we looked at a much, much more difficult technique, which was involved using in situ PCR. Our previous experiments with PCR, looking for DNA, HSV1 DNA in brain, involved extracting brain DNA and looking for specific sequences in the extracted DNA, which obviously consisted almost entirely of human DNA, but with tiny traces of HSV1 DNA sequences, which we picked up by using several primers for HSV1 DNA specifically. In this case, what you look at is slides, tissue sections of material post-mortem. And in many ways, although it's much more difficult to set up, you can argue that this has far fewer possible artifacts than solution PCR, although it, of course, has its own artifacts, the usual way that no technique is perfect. And what the top left shows HSV1 DNA stained by in situ PCR, and the middle one shows the plaques stained by a stain called thioflavin S, which green, and then the two together, merging them, shows exact co-localization. The lower picture shows material from another brain. In this case, the in situ PCR again is brown, but the staining for beta amyloid rather than for plaques as such, by staining for beta amyloid by immunohistochemistry, and it's a purple stain. I'm afraid it's rather hard to see. But what we could show from that, or what we found was, by quantifying it, semi-quantifying it, that HSV1 is present in most plaques in Alzheimer's disease brain, 70-something% it was, but in far fewer plaques in aged normal brain, where it was 20-something%. So vastly more of the HSV1 DNA is present in plaques in Alzheimer's disease brains. This was an exciting finding, and we were very pleased at that. So the answers to the four questions were, is HSV1 present in normal brains? And the answer to that is yes, it's present in the elderly, as well as in a high proportion also of Alzheimer's disease brains, but that does not mean it is not a risk. As I've said several times, infected does not necessarily mean affected. Secondly, is the virus active in brain? And the answer is yes, probably recurrently. In fact, there's been quite a bit of work, and I'm going to show a slide later showing that it is likely to be recurrent. Hence, it's likely to cause cumulative damage. Thirdly, is the virus in brain associated with Alzheimer's disease? And the answer is yes, in ApoE4 carriers. About 60% of cases are accounted for by HSV1 in brain of ApoE4 carriers. And also, as we found, ApoE4 is a risk for cold sores, which are usually caused by HSV1. So we can say that ApoE4 and HS carriage and HSV1 DNA in brain accounts for something like 60% of cases. As far as the other 40% is concerned, as it's almost certainly a multifactorial disease, we have no idea what they are. It could be bacterial. Several bacteria have been implicated after our first study looking, as I said before, for microbial DNA, in this case HSV1, the first paper we published was 1991. And a few years later, people produced some papers on various bacterial DNAs which they found in brain. So those could also be a risk factor. Then the fourth question was, is there a causal link with Alzheimer's neuropathology? And the answer is yes. HSV1 infected cells and infected mouse brains accumulate beta amyloid, and the infected cells accumulate AD-like tau. And from the in situ PCR study, HSV1 DNA resides in a high proportion of AD, Alzheimer's disease plaques, much lower proportion in elderly normal plaques. So what one can really draw, what this points to is that one should could or should use antiviral treatment to slow or possibly to stop HSV1 damage, HSV1-induced damage in brain. These all present strong support for this idea, and a lot of other publications more recently have supported that. And we, as I said before, we think it's not just an association, but a causal one. Much harder to show in human brain, because you can't just go and infect people with something and see if they develop Alzheimer's. And studies in mice, although they're quite suggestive and supportive, obviously mice are not humans. Anyway, what we did next was we infected cells in culture, and then we used a well-known antiviral agent, acyclovir, which has been used for some decades for people who develop this very, very serious, previously fatal disease, HSE, herpes simplex encephalitis. And it worked very well, although they're still usually affected, but they're not killed, which used to be the case for almost 100% of people who got encephalitis in the past. Horrible illness, horrible disease. So what we did here, we infected these VERO cells, and we looked, the top graph shows when we quantified using quantifying in quantified immunocytochemistry, the HSV1 proteins in the top graph, HSV1-induced amyloid accumulation, and abnormal phospho-tau in the bottom one. And what it shows is with increasing concentration of acyclovir, you get a corresponding decrease in level of viral proteins, viral-induced beta amyloid, and viral-induced phospho-tau, abnormal tau. And only in the case of the abnormal tau does the acyclovir at sufficiently high concentration reduce the abnormal tau to 0. In the case of the others, in the case of the viral proteins, it's reduced to 40%, and in the case of amyloid to just over 20%. Perfectly good explanations for those, which again I don't have time to go into. But what it really shows is there's a very large effect of acyclovir on these particular features of characteristics of Alzheimer's type brain, but in cell culture, infected cell cultures, production of amyloid and production of abnormal tau. So what's the conclusion to do with antivirals? Well, a conclusion is that acyclovir, we thought, combined with antivirals which inhibit the entry into cells of the virus, such as certain fucans, which are food supplements very popular in the Far East, which are made from algae or seaweeds particularly. The combination of the two, acyclovir, which inhibits viral DNA replication, and cell entry inhibitors such as these fucans, will be a very suitable combination for treating AD, because the fucans hopefully will stop most cell entry, and the Alzheimer's, the acyclovir will stop those viruses that have got in despite the fucans. But antivirals have the great advantage they would target only the virus-infected cells, not the host cells. And antivirals, acyclovir plus an entry inhibitor, should prevent most or all viral damage, irrespective of the role of beta amyloid or abnormal tau in Alzheimer's disease, about which there's a lot of discussion. So to summarize our results, HSV1 DNA is present in latent form but reactivates in brain of many elderly people. The resulting damage is probably limited and localized. In other words, possibly it's like one could say a mild form of herpes simplex encephalitis. It must be mild, the damage on each reactivation. Otherwise, the people would show symptoms of herpes simplex encephalitis, which luckily they don't do. I mean, the symptoms are fairly obvious. The next point is HSV1 DNA in brain, the presence of the virus in brain or viral DNA of ApoE4 carriers confers a strong risk of Alzheimer's disease. There's a lot of evidence that it's a risk and not an association alone, although there's no proof, I have to stress. Also, ApoE4 carriage is a risk for development of cold sores. As I said, the virus reactivates in everybody, almost certainly, but only some actually show symptoms. The cold sore sufferers are the ones who are symptomatic. The remaining 75 or whatever % are asymptomatic. HSV1 infection of human brain cells and of mouse brain causes Alzheimer's disease-like changes. And importantly, HSV1 DNA is located inside most plaques, amyloid plaques seen in Alzheimer's disease brain. And from that really sprung the idea that amyloid might be protective, at least at low levels and initially, because it looks as if it's sequestering or encaging the viral DNA. So I think this was the actual idea, as well as some suggestions in another paper by people, Steve Robinson and co-workers, who suggested that amyloid might in fact be an antimicrobial peptide some years, early 2003, I think it was. The next thing is antiviral treatment reduces greatly the accumulation of A beta and P tau in virus-treated infected cells. Then there are two things that I didn't have time to show, but are pretty important. We did a study in collaboration showing that vaccination of mice prevents HSV1 latency in brain, which was very interesting. Unfortunately, there's no good vaccine for humans yet. If there were, that would be an enormous advance, because vaccination prevention is far better than treatment. The other thing which I didn't have time to talk about, which is I think equally important, is that ApoE determines the severity of damage, or else perhaps susceptibility to infection, by a wide number of pathogens. There's a review on this, which some rather old now, but we found in a variety of diseases, ones in which ApoE entry of the protein use the same receptors as the actual pathogen, and we suggested that it might compete for entry into cells, but we haven't been able to prove that. But it's a very interesting result that it does seem to be a determinant. It influences severity of damage or susceptibility to infection. Anyway, perhaps I should just spell out the HSV1-AD concept. As we said, HSV1 DNA, the virus, resides latently in brain of many elderly people, and we suggest that it probably travels from the PNS to the brain in middle age as the immune system declines. We say that because we didn't find HSV1 DNA in a appreciable proportion of young people, only in a relatively very small proportion. And we're suggesting that the virus can reactivate possibly recurrently and then causes damage, which is almost certainly will be limited and localized, but it accumulates, eventually leading to Alzheimer's disease. So we then also found that HSV1 DNA and the genetic factor carriage of ApoE4 confers a strong risk, or certainly a strong association with Alzheimer's disease. We think it's a risk from evidence, and more evidence is coming out about this now. More damage probably occurs, we suggest, in ApoE4 carriers than in those with the other ApoE alleles. This is our explanation, a tentative one, which is why ApoE is the sort of cofactor, by the look of it, in causing damage leading to Alzheimer's. And that's really supported indirectly by the fact that we found ApoE4 carriage as a risk for herpes labialis, cold sores, in other words, for damage in the peripheral nervous system. And there are now well over 200 studies using very diverse methods, approaches by many other labs, including epidemiological, molecular, and cell biological, virological, and GWAS studies, which support HSV1's role in AD. What's the evidence, though, of this important concept of reactivation in brain? Well, firstly, our own study showing HSV1 antibodies are present in the cerebrospinal fluid of Alzheimer patients and age-matched normals. In other words, there is HSV1 DNA in brain, and it must have replicated, otherwise there wouldn't be antibodies to it. So its replication and expression have occurred in brain, probably recurrently. We showed in this paper, Wozniak et al., 2005. HSV1 DNA was detected in CSF of far more people than would be expected. This was a lab which looked at batches of CSF to look for HSV1 DNA. One would expect it to be present in a very low proportion indeed, because fortunately the HSE, herpes encephalitis, occurs only in a very small proportion of people, a few, 1 or 2 or 3% in a million. So you would not expect to find HSV1 DNA in the CSF of many people. In fact, they detected it in far more people than would have been expected. This suggests that something like mild HSE is actually going on in far more people than, in other words, Alzheimer's disease patients and elderly normals, which is what we found. And that suggests that virus reactivation is relatively frequent. And obviously, it's more harmful in those who develop Alzheimer's disease, possibly through carriage of APOE4. Then the next is rather long. I take too long to explain, so I'm going to have to leave it out. And the same with the last one about HSV1 infected mice. But both the third and the fourth point suggest that reactivation is far more efficient in immunodeficient mice and in immunodeficient humans. And this was done a study in 2015, subsequently substantiated by a study in 2016. There are also many case reports of mild or atypical herpes simplex encephalitis, and also many case reports of recurrent HSE. So these support also this idea of reactivation in brain. Just quickly going through lab members. On the left-hand side shows current lab members, and the right-hand side are collaborators. You can see there are very few. My group has always been very small due to unfortunate lack of money, very little funding for this sort of work, regarded as heretical. And just very briefly going through, there are now over 200 publications which directly or indirectly support a role for HSV1 in AD. I've said a variety of methods. Secondly, there are a number of results of others. This is just really going through the various methods they've used. I won't dwell on that because I've mentioned it, but measles virus causes neurofibrillary tangles and HIV causes amyloid plaques and neurofibrillary tangles. So other viruses can cause this sort of damage, but HS1 is the only relevant one found so far in elderly brains. Then there's some other studies here, which again I haven't got time to go through, but they all support, and the vast majority of these, well over 200 papers, support this HSV1 involvement in Alzheimer's. And there are a number of studies using bacteria which support bacterial role possibly as independently. I won't show this slide or the last one because there just isn't time. But thank you very much for listening, and I hope that you were able to enjoy the talk.